Good morning, Cellutopia. Today we're gonna have some very special guests. They're gonna be telling us about the process of cellular respiration. The first one will be glycolysis. Hello. It is said that you're the very beginning of the process. Is this true?
Of course that's true. I produce pyruvates which are used in anaerobic and aerobic processes.
Really?
Yep.
Is that so? Can you explain to me your job?
I break down glucose in the cytoplasm from 6-carbon to 3-carbon pyruvates with a net of 2 ATP.
Wow, that's amazing. Thank you for sharing that with us.
You're welcome.
Now, welcome. Fermentation. Can you explain to me your job?
Well, I create energy for the cell, or ATP, when oxygen is not available.
Can you explain to me the processes of fermentation?
Well, during fermentation, electrons are stripped off to regenerate the electron carrier, NAD+, from the NADH produced in glycolysis. CO2 and alcohol make yeast during alcoholic fermentation, and lactic acid is used to make dairy and helps keep you moving. When you're exercising and you're low on oxygen, this only makes less than 5 ATP though.
Is that why my muscles feel sore after I exercise after a long break?
Exactly.
Thank you, fermentation, for joining us.
You're welcome.
The Krebs cycle. Where do you occur?
I occur in the matrix of the mitochondria. I am also an aerobic process.
People say that you're like a taxi service. Can you explain me this analogy?
At first, 2 carbons created from glycolysis are added to a 4-carbon molecule. This is the building blocks of citric acid. 1 carbon is stripped and becomes a waste product. This is carbon dioxide. NAD+ turns into NADH. ADP turns into ATP. Another carbon is stripped. Next, the 4-carbon chain is rearranged to charge up FADH2.
What happens after that?
The cycle keeps continuing as the FADH+ and the NADH go on to be used in the electron transport chain.
Wow, thank you for joining us. Speaking of the electron transport chain, let's welcome them in.
Wow, it is such an honor to be here. The knowledge of the cell is such a passion of mine.
Can you simplify the complexity of the electron transport chain for us?
Yes, of course. I'm located within the inner membrane of the mitochondria. Electrons are transported from the NADH and the FADH2 to the electron carriers. This is used to create a proton gradient.
Wow, interesting. What else?
The electrons are used to power the ATP synthase proteins created from the protein gradient are used to power the ATP synthase as well. Hydrogen ions are diffused back through the cycle, so it spins and ATP are charged. Inside the ATP synthase, 1 phosphate is added to ADP to make it ATP. A maximum number of 34 to 38 ATP are made. Water is a waste product of this cycle.
So you're saying you're more efficient than fermentation?
Exactly. Thank you for having me today.
You're welcome. Thank you for watching us, Cellutopia. Signing off from the Dr.Cell show.